Hopefully you didn't see that. All right, can somebody confirm you can hear me?
I can hear you.
Thank you. Let me open up the grade book. Let me briefly talk about the grades. This weekend I worked on the grades. I got the extra credit study sheet graded, the extra credit picture graded, extra credit study 2 graded, the worksheet for lecture 2 graded, and I believe, except for one person, all of the quizzes, except for the last quiz, which you just took yesterday, are graded so that that's the plagiarism quiz 1, plagiarism quiz 2, the environmental quiz, practice quiz 1C is graded. Let me state that for practice quiz 1C, you can still see your answers and the quiz if you come into Canvas through the link on the main Canvas page. Let me briefly show that. I have to be careful because I've got the gradebook open, but I'm not showing that at the moment. There it is. Practice quiz 1B. If you click on this link, you can still get to the quiz, but I made this quiz not count in the grading, and so you cannot get to it to the gradebook. However, if you want to see your score, better stop sharing. If you want to see your score, you can go into your grades and you can see that I transferred your score into a separate column called Practice Quiz or QZ 1C, the C standing for corrected. Quiz 1C I've not graded yet. I've not checked to see if the scans were correct, and I'll try and get that done today or tomorrow, but I have not started at all on Quiz 1C yet. Any question about, I also have Lab 0 and Lab 1 are graded. They were only going to be graded one time. And then Lab 2 has been successfully graded the first time. If you did not get 5 out of 5 on Lab 2, you can submit the lab again for full credit by this Saturday. So 11:59 p.m. this Saturday, you can submit Lab 2 again to get 5 out of 5. And if you submit your answers again and you get it wrong, I'll mark it wrong and I'll tell you what the correct answer is. If you do not submit the quiz a second time, not the quiz, the lab a second time, and you want to see the answers, you'll have to send me an email. So if you only submitted it one time, I'm not going to give you the answers unless you request it in an email. And the reason is I just don't go back and grade it if I've already graded it. Any question about any of that? All right, let me close down quiz 1C. Let me close down the grade book. All right, let me share my screen now. Any question about anything? All right. So today is January 22nd. And we're supposed to be doing Lab 4 on stains. See, I made a mistake. Lecture 3 was not supposed to be finished today, so we do have a little bit more time for lecture 3. All right, let's go to the labs. I guess I can close that down, which I probably should do because the almost 95% done at finishing the lecture lesson. So my computer is working kind of slow, so that's why I closed it down. Anyways, lab 4, staining techniques, simple, differential, and special stains. You do need to read in the textbook. Read about the cell shape and arrangement of bacterial cells, only about 2 pages. Read about bacterial cell wall, also 2 to 3 pages. 80 to 83. Read about the simple stain, only a page. Read about differential stains. Overview will be 2 pages, 65 to 67. The gram stain is discussed on page 65 to 66, and the acid fast stain, the other differential stain, on page 66 to 67. Then read about special stains in general on page 67 to 68. The negative stain for viewing a capsule, the cell capsule, on page 67, as well as page 75 to 77. Then read about the endospore stain on page 68 and page 92 to 94. And then the flagella stain on page 68, also discussed on page 77 to 78. There are some video clips in the virtual lab in this lab, so view those. The learning objectives, you should, one, be able to prepare a bacteria smear for staining. And let me reiterate that when you make a bacteria smear, it is a separate and distinct procedure from doing the stain. The bacterial smear is not a part of the staining procedure. You just have to do the staining procedure on a bacterial smear, but you do not need to stain a bacterial smear. And then two, understand the principles and the procedures used when performing a simple stain, a differential stain, and a special stain. 3, interpret the results for a simple stain, a differential stain, and for special stains. And then 4, define these following terms. Any question about what we are going to be learning? All right. Bacteria cells are essentially transparent. Therefore, it is necessary to stain them before they can be observed, or at least observed well, under the microscope. You normally prepare a bacterial smear first, and how you do that is you add a drop of sterile water placed on a clean slide, and then you add a little bit of bacteria to the drop of water. How much bacteria? An amount that you can barely see on your loupe, meaning you do not put a loopful, you do not put half a loopful, you do not put a quarter of a loopful, you don't even put an eighth of a loopful. You have just barely enough you can see on your loupe, and then you add it to the drop of water. You then take the loupe and clean it by putting it in the Bacto incinerator or putting it in the flame, and then take the loupe, cool it, and put it in the drop of water and smear the drop of water all over the surface of the slide. You then allow the slide to air dry. Smearing out the drop of water will spread it out so it'll dry quicker. Isn't terribly important, but it does allow the slide to dry quicker. Next, you then allow the slide to air dry, and then the cells are heat fixed onto the slide by gentle heating. This will keep the cells from washing off the glass of the slide during the staining process. We'll talk about how you do that in just a little bit. In the simple stain technique, you use one dye to stain bacteria cells. Notice how I scratched the word basic dye because it doesn't matter if it's an acidic dye or a basic dye. You only use one dye and it is a simple staining technique. You can determine the cells by their shape and their arrangement, but not by their color because they'll all stain the same when you use one dye. A differential stain usually uses two or more dyes to provide different information about the cell. And lastly, we have special stains, which are used to dye a particular bacterial structure, such as the cell capsule, the cell's endospores, or there's only one endospore in a cell, but if there's more cells, you can get more endospores, or the flagellum. So each of these would be a special stain to either view the capsule, the endospore, or the cell's flagella. When you stain cells, you can note the growth pattern of the cells, also called the cellular growth arrangement or just the cell arrangement. You can have, first of all, single cocci, which is not shown here. You can have diplococci, that's two cocci stuck together. You can have a tetrad of cocci, shown here. You might have a sarxinia, that's 8 cocci stuck together. You can have streptococci, that's cocci strung together in a string. And then you can have staphylococci, a cluster looking like grapes. With bacilli, you have some of the same cellular growth arrangements, but they are fewer. You do have single bacilli, you do have diplobacilli, you have streptobacilli. You do not have staphylobacilli because that doesn't exist. I don't think you have tetrad bacilli either. You can also tell besides the cell growth arrangement or the cellular arrangement, you can also determine the shape of the cells. You could see bacilli, you could see cocci, you could see coccobacilli, that's a shape in between a cocci and a bacilli. You could see a spirilla. And these are the three main shapes, the bacilli, the cocci, and the spirilla. Coccobacilli is in between the bacilli and the cocci. And then the cells could be pleomorphic, where each cell takes a different shape. Most cells are not pleomorphic, but some species are. Any question about the simple stain? There's also the differential stain invented by Hans Christian Graham. You don't need to know the year he invented it, but you should know the Graham stain was invented by Dr. Grahams. And so you should always capitalize the G in the Graham stain. The Graham stain distinguishes between two very large group of bacteria according to how the cell retains the crystal violet. Bacteria that retain the crystal violet after the staining process are classified as gram-positive, and these cells appear purple, while bacteria that lose the crystal violet are referred as gram-negative, and these cells appear pink, as shown here. We have, let me blow this up a little more. We have gram-positive cocci right there, gram-positive bacilli right there, gram-negative bacillus right there, and a gram-negative spirillum right there. So the gram-positive cells appear pink, the gram-negative appear pink. The color of the gram cells, meaning the gram-stained cells, depend upon the cell wall composition of the cells. The gram-positive bacteria have a thick, structurally different cell wall, which has a very thick peptidoglycan layer, at least 30 layers of peptidoglycan, up to hundreds of layers of peptidoglycan thick. The gram-negative cell walls have a different cell wall structure. The number one thing is they have a thinner peptidoglycan layer, between 3 layers to about 20 layers of peptidoglycan. And actually here you can see, if I blow it up a little, you can see that this one has several layers of peptidoglycan. There's one layer there, there's 2, 3, 4, 5, 6, probably 7, 8, 9, 9, 10, or 11, something like that, layers of peptidoglycan shown here. The gram-negative cell also differs from the gram-positive cell, and that is peptidoglycan is by far the largest, most common molecule in the cell wall. There are a few other molecules, but peptidoglycan makes up the vast majority of the cell wall in a gram-positive cell. Very thick layer of peptidoglycan. In the gram-negative cell wall, peptidoglycan is the main molecule in the cell wall, but it does not make up the vast majority of the cell wall. There are other molecules in the cell wall, included this lipid layer, which has other molecules like proteins and carbohydrates in it. But there are other molecules in the gram-negative cell, mainly this lipid bilayer, which we call the outer membrane of the cell wall. So it is part of the outer membrane, but it is a membrane itself. It's part of the cell wall, sorry, but it is a membrane itself, and it's called the outer membrane of the cell wall. Any question about any of that? So the gram-positive cell is mainly peptidoglycan, which are polar molecules. Water can go right through the peptidoglycan layer. The gram-negative cell wall does have that peptidoglycan layer, but it has an outer membrane, which is lipid. And a water-soluble molecule has to go through 2 membranes to get inside the gram-negative cell. It has to go through the outer membrane as well as the cell membrane to get inside the cell. Whereas another molecule coming through the gram-positive cell has only to go through the plasma membrane to get inside the cell. Any questions about the difference between the gram-negative cells and the gram-positive cells? All right, in the gram stain, after bacteria have been stained with crystal violet, Gram's iodine is used as a mordant, which this iodine chemically combines with the crystal violet to form the crystal violet iodine complex. That complex is larger than crystal violet or iodine alone. In the gram stain, we then have the decolorizing step, usually using alcohol acetone, which is applied briefly to remove the excess crystal violet from the cells. It'll pull all of the crystal violet iodine complex out of the gram-negative cells, but not pull out the crystal violet iodine complex from the gram-positive cells due to the thickness of that peptidoglycan layer, meaning the crystal violet iodine complex gets stuck in the peptidoglycan layer, does not wash out. In the thinner peptidoglycan layer of the gram-negative cells, the decolorizing step pulls out the crystal violet iodine complex. Any question about any of that? So the timing of applying the decolorizer is critical. And I'll talk a little bit more about that in just a minute. So gram-negative bacteria are colorless after the decolorizing step. And then to give them color so they're easier to view, we counterstain them with the dye safranin, which is pink in color, giving us the gram-negative cells and the gram-positive cells, staining purple, the gram-negative staining pink. Any question about any of that? All right. This slide here is actually not showing you gram-negative and gram-positive cells. They're staining that way, but this is a gram variable result, meaning only one species is on this slide. And yet the cells are staining both gram-negative and gram-positive. What do you notice that's different between this cell here and that cell there, and then these two cells here? What do you notice that's different about them? Anyone? Come on, the worst you can be is wrong. And actually, it's pretty obvious.
The color or the shape?
A little bit of both. Meaning, what's different between this cell and these two cells? Besides the color pink and the color purple, what else is different?
They're clumped together.
Well, this one is clumped together with that one, and that one is clumped together with three. I agree with that, but that's not what I was getting at. You see a break in that cell right there? The boundary of this cell is not distinct, and then it looks like the cell is even broken right there. And this cell and that cell have very distinct boundaries. There's no break in the cells. And then this cell there, that cell looks like a ghost. Up to the pointer, top of the pointer, that cell there looks like a ghost. But this part right up there, the cell, you can see through a part of it. So it looks like a ghost. Probably the reason why these cells are gram variable is because this cell is dead and that cell is dead. Whereas these cells are viable. And my guess is a lot of these pink cells are dead cells, and most of these, if not all of these purple cells, are viable cells. And that's why the purple cells have distinct boundaries, and the pink cells do not always have a distinct boundary. The cell can be broken, or the cell looks like a ghost. It's because the cell is dead. Why the cells are dead and why there's so many dead, if the pink ones are all dead, for example, and the purple ones are viable, is because these are too old of cells. To do the gram stain correctly, the cells have to be no older than 2 days old. All the gram variable results I've ever seen, when students have done the slide and made on one species and got gram variable results, I asked them, how old are your cells? And everyone told me, oh, these cells are older than 2 weeks old. They're old. And I told them, okay, to do the gram stain correctly, the cells should only be 2 days old or younger. If you're using old cells, you'll have dead cells in them, and the dead cells may not stain correctly. In this case, the cells aren't staining correctly because the cells are too old. They're dead, and then the cell wall is not complete. It's got holes in it, something like that. That clearly has a break in it. And that clearly is a dead cell because it's a ghost. And because there are holes in the cell wall, the crystal violet iodine complex can wash out. So you should never do the gram stain on cells that are older than 2 days old, because if there are lots of dead cells, they may stain gram negative, as this slide here shows. Usually, the gram variable results only shows up on the mycobacteria cells that are old. If you stain other old cells, usually they all stain either gram-negative or gram-positive. But with a mycobacteria, you can get gram-variable if you stain old cells. Any question about any of that? Why the mycobacteria tend to stain more gram-variable, especially when you're using old cells and other cells, is because the mycobacteria have a lot of mycolic acid in their cell wall, and that prevents water-soluble cells, water-soluble stain from even getting inside the cell. And usually the mycobacteria, they do stain gram-positive, but they stain lighter than other cells that lack the mycolic acid in their cell wall. And that's only if you use freshly grown mycobacteria. If you use old mycobacteria, they will frequently stain gram-variable, like this slide here. And all the gram-variable cells I've ever seen have only been mycobacteria that were 2 weeks old or older. So some tips to prevent getting gram-variable staining procedure: avoid using old cells. The cells should be 2 days old or younger, and if they're not, you should streak out the cells to get fresh growth and then do the gram stain on freshly grown cells. You should prepare thin bacterial smears. If you have a thick bacterial smear, you'll have trouble getting the dye all the way to the bottom cell. And then if the cell is gram-negative and the layer is too thick, you'll have trouble getting all of the crystal violet iodine complex out of many layers of bacteria. You should heat fix the bacteria only after the smear is air dried. If you do not air dry the cells and then you heat fix the cells, you'll get water boiling inside the cell, and that can put holes in the cell wall, which can then throw off the gram stain. You should also avoid overheat fixing the cells. Usually if you overheat fix the cells, you'll note the cells looking melted, but if they look melted, you've overheated the cell and you may have damaged the cell wall, so the gram stain may not stain the cell correctly. And then use fresh staining reagents. Do not over decolorize, do not under decolorize. Any question about any of that? All right, the next differential stain we're going to talk about is the acid-fast stain. Some bacteria, specifically the Mycobacterium, but also the Nocardia, have a waxy substance inside their cell wall that is mycolic acid. These bacteria are able to resist acid-alcohol decolorization, and so these bacteria are called acid-fast bacteria. The acid-fast cells retain the acid-fast stain, and then the acid-fast negative cells lose the acid-fast stain. Initially, all cells take up the primary stain, which is called ball fusion. It's a cherry red color, sort of a pinkish red color. And then the cells are decolorized with acid alcohol. That will pull out the carbol fusion from the acid-fast negative cells, but not from the acid-fast positive bacteria. And then a basic dye is applied, usually either methylene blue or Belgian green. And it's applied as a counter stain, simply to stain the cells that have been decolorized. This will give the cells a different color if they're acid-fast negative. If methylene blue is used, the acid-fast negative cells will be blue, but if brilliant green is used, the acid-fast negative cells will be staining green. So here we're looking at some acid-fast bacteria as well as acid-fast negative bacteria. If the cell retains the pinkish color or reddish color, it's an acid-fast bacteria cell, likely Mycobacterium or Nocardia, simply because they contain the mycolic acid in their cell wall. And then the acid-fast negative bacteria stain, in this case, a bluish greenish color, depending on whether you're using differential green or brilliant green or methylene blue. I think this is methylene blue because it's kind of a bluish color. Any question about any of that? All right, you may have noticed here I've mentioned that the Nocardia has mycolic acid in it as well as mycobacteria. The mycobacteria have more mycolic acid in it, which is why when I first talked about mycolic acid, I only discussed the mycobacteria. The Nocardia have enough mycolic acid to remain an acid-fast positive bacteria, but they do not have enough mycolic acid in the cell wall to prevent the gram stain from staining the cell, or also generally they don't have enough mycolic acid to make the cell gram variable. All right, let's talk about the special stains. Any questions about the differential stains? Remember, there's only two differential stains you need to know. The gram stain, which you should know the procedure of that stain, and then the other is the acid-fast stain. Let's talk about the special stains. There are going to be three that we're going to talk about. A negative stain for staining glycocalyx. Remember, a negative stain is just a negative stain is a acid stain as opposed to a basic stain. It stains this stain, negative stain, we're going to look at it for staining the glycocalyx, also called the cell capsule, if you can see it under the light microscope. And then the second special stain we're going to look at is the endospore stain for staining endospores. And then the third special stain we're going to look at is the flagella stain for staining flagellum or flagella. All right, the glycocalyx can be attached firmly to the cell wall, and if it's thick and you can see it under the light microscope, we call this glycocalyx a cell capsule. Production of a capsule can be associated with an organism's virulence or its pathogenicity, meaning that if a cell has a cell capsule and you get the infection, the cell will be more virulent or more likely to cause a disease or worse disease than a bacteria that lacks a cell capsule. The cell capsule helps prevent the bacteria from being phagocytized by white blood cells. And that's why if a patient gets an infection with Streptococcus pyogenes causing strep throat, most of the time, not all the time, but much of the time, the patient will not be able to throw off the bacterial infection because the bacteria contain a cell capsule. The cell capsule the bacteria colonies look mucoid. So the reason why these bacteria look mucoid is because they have a cell capsule around each cell. Capsules are not easily observed using conventional staining methods. Like when you apply the gram stain, not only will the cell capsule not show up, but the gram stain will generally wash off the cell capsule from the cell. So therefore, we frequently use a negative staining technique to show the presence of a cell capsule. India ink or nigrosin are commonly used as a negative dye to display the cell capsule. This is using nigrosin, I think, and one other stain to show the cell capsule. This is two stains. One is staining the bacteria cell, and then the other stain is studying, probably the nigrosin, is staining the background of the cell. And you can see the capsules do not stain around the cells. Here we're using India ink, one stain, which stains the background of the cell very dark, almost black, and it lightly stains the cells, kind of a grayish color. Once again, the capsule does not stain with India ink, and you can easily see the capsule from the background as well as from the cell itself. Any questions about a negative capsule stain? All right, let me state that we do have different types of capsules, different types of glycocalyxes. I'm not going to talk about in this lab, but the main other glycocalyx that is not a capsule is a slime layer. A slime layer is mostly water. It's loosely attached to the cell wall, and you cannot see the slime layer with a light microscope because it's mostly water. Whereas the capsule has structure to it, other molecules in it. It too is mostly water, but it does have other molecules in it, so that I don't know, there's a significant number of other molecules in the cell capsule. The majority is still water, but in a slime layer, over 95% of the molecules in the slime layer are water. And it might be over 99%. It's very high. All right, any question about capsules? If not, let's move on to the endospore stain. Endospores are metabolically inactive structures inside a cell. They are hardy and are produced by some bacteria as a defense strategy to survive unfavorable environmental conditions. When the environment then becomes favorable, the endospore can germinate and return to a normal metabolically active cell that we call a vegetative state, or the parent cell, one that is metabolically active, capable of reproducing, whereas the endospore is not very metabolically active. I'm not saying it's going to have no metabolic metabolism, but essentially none. And it is, well, it's not dividing. The endospores tend to be most common in two genera, Bacillus and Clostridium. So most of the endospores are in the genus Bacillus or the genus Clostridium. There are only two other genera, I guess, that contain endospores, and I think both of those other two only have one species in it. All right, this table is showing you some of the pathogenic endospore forming gram-positive bacilli. We have Bacillus anthracis, which causes the disease anthrax. We have the bacteria Bacillus cereus, which causes food poisoning, especially food poisoning in rice is oftentimes caused by Bacillus cereus. So there are some anaerobic bacteria in the family Clostridium, like Clostridium botulinum, causing food poisoning, aka botulism. Clostridium tetani, causing tetanus, also called lockjaw. Clostridium perfringens, causing gangrene and food poisoning. C. perfringens, no, it's Clostridium difficile, causing antibiotic-associated colitis or pseudomembranous colitis, most commonly called C. diff, kind of common in hospital-like settings because the endospore can survive anywhere. And if you have a patient receiving antibiotics, it will not kill out the endospore, and then the Clostridium difficile can overgrow in the gut because it has no competition, and then it can cause antibiotic-associated colitis. All right, the endospore stain is sometimes called the Schaefer-Fulton endospore stain. It uses malachite green as the primary stain. Heat is then applied as a mordant. The cells are then colorized with water, which will remove the malachite green from a vegetative or a parent cell, but not from inside the endospore. Safranin is then applied as a counterstain to stain any cells that have been decolorized so that a vegetative cell will appear pink and then the endospore will appear greenish. Let me blow this up. Here we have a parent cell, pink. Call it a vegetative cell as well. Here we have endospores which would come out of the cell, and these endospores are different shape from the parent cell. They're kind of coccoid, coccobacillus, and they're a different color, obviously, green. And here, let me blow this up even further. Here we have an endospore being formed inside of this parent cell. Same with that one. And that's actually where the term endospore comes from, meaning a spore inside the cell, endospore. Once the parent cell finishes productive of the endospore, the parent cell will eventually die because of deteriorating environment. And once it dies, it will then release the endospore from the parent cell. Any question about either the endospore stain or the endospores? All right, let's move on to our third and last differential stain, the flagella stain. Come on. Flagella provides bacteria with the ability to move. So if the bacteria or the archaea can move or swim in broth or water, they have flagella. Why can I say that? Because the flagella is the only structure that both the bacteria and the archaea have for locomotion. That's not true in eukaryotes, but for bacteria, the only structure they can have for moving about is the flagella. Flagella are composed of flagellin. This is a class of proteins. You can differentiate the cells on whether they have flagella or whether they do not. But you can also differentiate the flagella for where the flagella are on the bacterial surface. Like monotrichus, there's one flagella on one end of the bacteria. And with this cell, it beats sort of like a whip here, and that will propel the cell forward. You can also have bacteria that are lophotrichous, meaning they have more than one flagella on one end of the cell. You can have bacteria which are amphitrichous, and that is these bacteria have flagella on both ends of the cells, usually more than one, but at least one on both ends of the cells. Amphitrichous. And then we have cells which are peritrichous. These have flagella all over the cell. You need to know this only for the lab. I will not ask you questions about the flagella arrangement on the quizzes or the final. But you do need to know it for our lab today. All right, because bacteria are too thin to be seen using standard staining techniques, we have special stains that are required to view the bacteria flagella under the microscope. And here is some flagella. Seen under the microscope after the flagella stain. This cell has a lot of flagella on it. That cell has flagella mainly on this one end, so that would be amphitrichous. No, lophotrichous. That cell may only have one, but it might have two on one end. That cell looks like it has no flagella attached to it. And that cell looks like it has one flagella attached to it. So when we see cells of one species that have more than one flagella arrangement, how do we classify these species? Anyone want to take a guess? I'll give you extra credit if you guess right. All right. Whenever you see the flagella arrangement, you'll always see more than one flagella arrangement. Because initially, when the cell is first born, or first created, whatever term you want to use, the cell will have no flagella. But then the cell will start making flagellum, and they'll first come out in one, and it'll be monotrichous. Then multiple flagella may be made, and it'll be lophotrichous. And so you'll see both none, both monotrichous, both lophotrichous. You can also see cells which are all three of these, and then they're amphitrichous. And you can also see cells which are all four of these, meaning no flagella, no flagella, monotrichous flagella, lophotrichous flagella, and amphitrichous flagella, as well as flagella all over the cell, and these will be peritrichous. On this slide here, I gave you, the slide is made up of one species of bacteria, but yet we always see the flagella arrangement being pleomorphic because it takes time for the flagella to be made and put outside the cell. But if you had enough time, enough photographs, you'd eventually see all of these cells in this slide, unless it's dead, become peritrichous, regardless of what flagella arrangement it is beforehand. Any question about any of that? All right, know the terms of this lab? No, a special stain. You can look at the references if you like, and then do the lab 4 exercises in your worksheet. Everything you do will be done using online techniques, but you must understand how to prepare a bacterial smear and then staining that bacterial smear later for a staining procedure. And you should know the principles and the procedures used in these various techniques, as well as interpret the results using a microscope. So in step 1, we're going to prepare a bacterial smear, watch a video on it, and then watch a little video on heat fixing the bacteria to the slide. I'll let you read through procedure 1 and then answer the questions. So this is procedure 1, B1. Question number 3 has a question mark. So anything that has a question mark or a blank, you have to answer. What are the 4 common errors when preparing a bacterial smear for staining? So you need to list the 4 errors that are common that people sometimes make when preparing a bacterial smear to be stained. For procedure 2, we're looking at a simple stain. And 2A1, what is the cellular morphology, the shape, not the cellular growth arrangement, of 1 E. coli? And let me state that this picture is a bad picture. The cells tend to be clumped together. Let me blow this up a little. The cells tend to be clumped together, but if you look, that is not a half a spiral. There's actually 2 cells there, one here and one there. This one is clearly 2 cells, so that's not a 7, that's a 2 cells stuck together. And this one right here could be 1, 2, 3, 4 different cells stuck together. It may only be 3, but it's definitely more than 1 cell stuck together. So when you're talking about the shape of E. coli, you should be careful. None of them are spirillum. And then tell me the shape of the Staphylococcus aureus shown on the right. And then answer question 2B. What is the total magnification of the microscope we're using in this question? Six. What is the total magnification? We have a video clip. Oh, that's the 2B, sorry. Procedure 3 is on the gram stain. We have a video clip for you to watch on the gram stain. So watch that, and then open the virtual lab of the gram stain for procedure 3 and work through the virtual lab. I'm going to quickly move through the first couple of slides to get to where the gram stain actually begins.
Hi, lab assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria. All yogurt. This is what plain. Now take this yogurt. Let's collect in the grams. We're going. All right. To prevent bacteria. You're going to use a striker to light the Bunsen burner. The gas is turned on low already, So bring the
Virtual lab?
striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
When there's a arrow there, you have to start up here and then bring it down, and then that moves
We
on.
need to dilute the yogurt before. Start by placing 1 milliliter of the diluent in. This is an inoculation. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. After it has cooled down, use the loop. Place the yogurt sample into the previously measured diluent, PBS. Now, use the inoculation. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. Transfer a small amount of sample from the test tube to your prepared slide. Place 2 loops full from the sample onto the slide. Tilt the slide to spread the drop out slightly, about the size of a penny.
You actually want it to go more than that so it'll dry quicker, so you can spread it out almost over the entire surface of the slide.
Now we'll heat fix the slide. Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Be careful not to put it directly in the flame. We don't want to burn the sample. Just dry it out so bacteria won't wash off.
It's actually better to hold it
Now
with
we
your-
begin the staining process. After the staining is complete, we can look at these slides using a microscope.
In the last procedure, it's actually better to hold the slide with your hands and then put it into the flame, because if you get it too hot, the slide too hot, you'll start to melt the cells. And if you're holding it with your hands, you will not get it too hot because it would burn your fingers. So hold it in your hand and then put the slide into the Bunsen burner or the flame to gently warm it. You just want it to be a little warm. You do not want the temperature to be of the slide to be really hot. And that's why it's best if you hold the slide with your fingers instead of holding it with the slide holder.
We use 4 chemicals. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds.
So you can spread that all over the slide if you have cells all over the slide. After 60 seconds,
Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent. Squeeze several drops onto the sample, enough to cover it completely. Let that sit for 60 seconds.
Anyways, that's how you do the virtual lab on the Gram stain. So do that. And then review the steps for performing a Gram stain. First, you apply the crystal violet for 1 minute. It will stain all the cells, gram-negative and gram-positive, bluish purple. Then wash. You don't need to put that in every step, but after every step you do a wash gently with cold water. And then you add the iodine, the mordant. It will stain the cells purple color. And then you dump off the iodine wash. And then you do the decolorization step. Here we're using alcohol or acetone alcohol wash. And acetone alcohol? Yeah, acetone alcohol. And you only apply it by about 5 seconds. As I stated, the best thing to do is to hold the slide up to your eye, hold it at an angle, put the decolorizing agent on the top so it runs down. Once the blue stops running, you've known you've added enough decolorizing agent. So stop adding the colorizing agent, dump it off, and then quickly wash the cells with cold water, gently, but with cold water. And then wash the cold water off, apply the safranin, and now the cells that are gram-negative will stain pink from the safranin. The gram-positive cells retain the crystal violet and they stain bluish. Any question about any of that? Then fill in this table. E. coli, state what its gram reaction is, gram-positive or gram-negative, and then state the cellular morphology, the shape. And in this picture, the cells are better. There's less clumping. Do I have another picture? Oh, here it is. So the cells don't appear pleomorphic. Anyway, state the growth arrangement of the bacilli. No, you don't even need that. You just need to put the whether the cell is a coccus or a bacillus. And then do the same for Staphylococcus aureus. Give the RAM reaction, whether the cell was a coccus or a bacillus. And there's the organism there. Then in procedure 3, answer questions A, B, C, D, E, and F. This will be the color of a cell at various steps in the gram stain. Staphylococcus A is Staphylococcus aureus. What color is it after the primary stain is added? And your options have to be pink, purple, or colorless. And then E. coli, what is its stain after the primary stain is added? For procedure 3C, Staphylococcus aureus, what color was it after the alcohol acetone step? Procedure 3D, E. coli, what color was it after the alcohol acetone step? And then procedure 3E, Staphylococcus aureus, what color was it after applying the counter stain? And then procedure 3, step 4, F, sorry, E. coli, what color was it after the counter stain is added? Any question about any of these? Procedure 4 is the acid-fast bacterial stain. We have a video clip for you to watch on staining the cells with the acid-fast stain. We're going to review the steps using the modified Kenyon acid-fast stain. You first apply carbol fusion. It will stain all cells reddish or pinkish. You then wash that off and then apply the acid alcohol wash, removing the decolorizer from the acid fast negative cells, not removing the decolorizing, moving the carbol fusion from the acid fast negative cells, but not removing it from the acid fast bacteria, positive bacteria cells. So this is the differential step of this stain because the cells are now distinguished in between the pinkish acid fast negative cells and the clear acid fast. I said that wrong. The pinkish cells are the acid fast positive bacteria. The clear cells are the acid fast negative bacteria cells. Then you add the counter stain, which in this case is brilliant green. It will not affect the color of the acid fast positive bacteria. They'll still be staining green, pink. And then the acid fast negative cells will take in the brilliant green and take on the green color. Any question about that procedure? Shrink that down a little. For procedure 4, you must answer the questions denoted by the arrows. State whether the cell is acid-fast positive bacteria or if the cell is an acid-fast negative bacteria. If the cell is pink, it's acid-fast positive. If the cell is greenish or bluish, it will be acid-fast negative. In the modified Kenyon method, it's only greenish. So state what that one is, state what that one is, state what that one is, state what that one is. You can put your answer above the arrow, or you could say, right here, write it down. The first arrow on the left is state what it is, and then the second arrow on the left is state what it is. For this one here, say for the modified Kenyon cold method, or the second picture, the first arrow on the left is state what it is, and then the arrow on the right is this one, or you could say the second arrow on the left is that one, and then state what it is. Any question about answering that question? So make sure you answer the arrows for what they are. For procedure 5, we're looking at video clips for the endospore stain and the negative stain. We have 3 pictures. Enter procedure 5. For procedure 5, is that figure 5? Procedure 5, step 2, we have slide A, and it tells you that this is the Schaefer-Fulton stain, which uses what stain is the primary stain, and it ultimately dyes what cell structure, which is produced by some bacteria to aid in survival. So tell me what the primary stain is and what the cell is staining, or this dye is staining. And you can look that up by saying what the Schaefer-Fulton stain is, which of the 3 stains it is. And then we have procedure 5, figure B, and the questions to it are right here. And I guess I forgot to mention that this would be P5, A1, A2, A3, and you need to mention for A3, what are some examples of pathogenic endospore producing bacteria? You can get that in the table of the lab module as well as the text of the lab module. So for procedure 5, figure B, this is what staining these cells that coats long whip-like appendages which aid in motility. So state what either the stain is or what it's staining, and then state the flagella arrangement seen in this slide. So that would be B1, B2, and then B3. Answer, what are some examples of clinically significant bacteria that produce flagella? And you can get that in the lab module as well as online. All right, so for figure C shown here, we have question C down here. And it's saying, what type of a stain is this using India ink or nigrosin, and can be used to detect the presence of what cell structure, which is located exterior to the cell wall and prevents phagocytosis. So you need to name the specific type of stain. You can't say a special stain because that would apply to all 3 of these, but you can tell me what either the name of the stain is or the type of stain. For C1, for C2, name the structure of the cell that this stain is trying to illustrate. And then for question C3, answer what are some pathogenic microorganisms that produce a cell capsule? And let me warn you, when it's saying some pathogenic microorganisms, that is plural, that means that one answer is not going to be accepted. You only get partial credit. To get full credit, you must name at least 2 species, because one is not some, and microorganisms is not one microorganism. You must have 2 to answer that for full credit. And then answer the case studies. Case study 1 is a 45-year-old female experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray, which is shown here. And in case you don't know what a chest x-ray normally looks like, this chest x-ray looks really bad right there, and right there. It looks more normal down here. The chest x-ray should be mostly dark from clear space in the lungs. And obviously that's not mostly dark right there. So there's something showing up in the chest x-rays. They then did a sputum for an acid-fast bacteria culture. So the acid-fast bacteria smear was made and stained. And then here it is here. And that's with the Zeal-Nielsen stain. And what is the most probable diagnosis? To answer this question for full credit, you can't tell me the disease, but you need to tell me what species is likely causing this disease. For case study 2, we have a 64-year-old diabetic patient. He's admitted to the hospital with soft tissue necrosis of the foot, and that looks really bad. A routine culture and gram stain is ordered. The bacteria grew aerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. So here we're looking at the soft tissue necrosis. There we're looking at the gram stain. Here we're looking at the Schaefer-Fulton stain. You need to look and see what the Schaefer-Fulton stain is showing, and you can actually see it right there. Based upon the information you have, name the species likely causing this infection. Any question about the case studies? The case studies are thinking questions, so you cannot directly get the answer directly out of the lab module, but you should be able to get everything you need to be able to get your answer from using the lab module. I'll warn you, if you try and get AI to answer the question for you, it may give you an incorrect answer, because I'm only going to accept an answer that came from this current lab module. If you get another answer, you know that answer is going to be wrong because that species was not mentioned in the lab module. And another reason for not using AI is if you use AI and then I give you a quiz question, which is a case study, you will not be able to use AI to answer that question. So you should attempt to answer the question without using AI. And after you have an answer, you can then use AI if you want to verify your answer, to check and see if your answer is correct or not. But I do not recommend you use AI initially, because I've had students do it, and then when it comes to an exam case study, they have trouble answering the case study. Any question about any of that? All right, remember in the case studies, you need to answer a species causing the infection. You cannot name a disease. Any question about any of that? If not, I'll be here until 8:15 or the last student logs out to help you with any questions. Remember, I'm here for you to take advantage of me or to use me if you like. Of course, if you can do it on your own, that's perfectly fine too. But I will be here until 8:15 to answer any questions. All right, any questions? If not, go ahead and get started on the lab. I will be here until 8:15 or until after the last student logs out. Once the last student logs out, I will log out as well.